My name is Ben Mackenzie, and I'm a member of Team Heartbreak. We built a tool that addresses the rising costs of pharmaceutical drug development. This trend is driven in part by the need to validate existing research, which is usually done by reproducing expensive experiments. Our idea is that some research can be validated computationally, and we've built a tool to do that within the limited scope of research involving gene interactions. Our idea is new because while computational tools that support experimentally driven research validation exist, no end-to-end solution exists for validating research directly and computationally. The tool's pipeline starts with text processing, where a PDF research paper is parsed with Apache Tika and then tokenized into sentences and words using Python NLTK. The tool filters for tokenized sentences containing potential interactions, which are then converted into features and fed to a linear regression classifier. A set of gene interactions is created from the positively classified sentences. This set is compared to a larger set of the mine gene's first-degree neighbors from the string database, and the similarity of the sets is expressed using the Jacquard index. A combined graph of the networks that visually categorizes interactions is then created using D3. Interactions from the paper that are unknown to string are marked in red. Drug researchers can decide if these are new interactions worth exploring or mistakes that invalidate the paper. Interactions from the paper that are known to string are marked in blue and distinguished from those in string that are absent in the paper. This provides context for researchers and helps them decide how consistent the paper is with other research. The tool has two parts that needed to be tested experimentally: gene detection during sentence filtering and the classification of sentences that contain potential interactions. The gene detector, built using a simple comparison algorithm from a list of over 20,000 human genes, identified over 90% of the genes and papers we tested it on. The classifier's test accuracy was 98% after we trained and tested it on the BioC BioGrid database, but it demonstrated overfitting when it misclassified even simple manually created examples. The BioGrid dataset was abandoned and a custom set of over 200 manually annotated examples was created in the form of tokenized sentences. The classifier trained on the custom dataset achieved 58% accuracy when it was tested on the BioGrid database, showing better generalization than the first model. Despite the small size of the custom dataset, the second classifier compared well against other methods for extracting gene interactions from texts. It almost matched the scores for deep dive and grammatical relationships graphing, and only Bayesian inference beat it soundly. Our tool's performance would improve significantly with an improved classifier, which could be achieved if we had more high-quality training data. With more data, our tool could become a viable resource for validating research and lowering the costs of drug development.